You're staring now at your tech transfer timeline. Leadership wants aggressive dates. The CDMO is confident but vague, and you are carrying the weight of knowing that if this slips, then the entire program slips. Do you feel that pressure? Yes, it's real. The complexity also real. But here is also what's real. Tech transfer isn't magic. It's not an art. In part 1, you learned the 6-pillar framework. Now I'm giving you the implementation playbook, starting with the human element that most people completely miss. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Let me share this story. I was leading a tech transfer to a new manufacturing site. Everything was progressing, slowly but progressing. Except for one guy in QC. Every interaction with him was difficult. Analysis requests were unpredictable. My team was stressed, and his team seemed also stressed. And we had a few conflicts because the walls were just going up and up. I was convinced he was the problem. Resistant to change, maybe territorial about his lab. Then I did something I should have done weeks earlier. I walked into his lab. No agenda, just a conversation. And what I realized that day is that he wasn't resistant. He was drowning. His lab was understaffed. His backlog was crushing. And every time we showed up with the quote-unquote urgent sample request, it threw his entire week into chaos. He craved predictability. I was creating chaos. Once I understood that, everything changed. We built a sampling calendar that gave him some predictability. And I explained him also that even though I tried hard, I could not always plan everything. And he understood. The transformation was immediate, not because the science changed, because I finally understood what he needed. And here's the lesson. Understanding stakeholder needs is as critical as understanding the process. How do you manage your stakeholders? Let me give you your stakeholder protocol. First, map early. Identify every stakeholder. QC, quality assurance, manufacturing ops, process development, regulatory, even the facilities or your IT, if your process has specific requirements. Second, understand motivations. What does each stakeholder need? What are their fears? The QC manager might fear the analytical backlog. The QA manager might fear audit findings. The manufacturing supervisor might fear unproven procedures. Write it down. Third, create a communication plan. Match the frequency and methods to stakeholder type. Some people want weekly written updates. Some want face-to-face check-ins every 2 weeks. Some just want to know you'll call them if there is a problem. Fourth, use the power interest grid. Focus your energy on high-power, high-interest stakeholders. They're the ones who can kill your project or accelerate it. Fifth, Build trust face-to-face whenever possible, especially for the resistors. Email doesn't build trust. Video calls can be better than nothing, but nothing beats sitting in someone's space and listening. On another tech transfer, we transferred the process to a completely new site. We used the same bioreactor models, the identical SOPs. We'd even sent people for hands-on-hands training. But then something surprising happened. We produced the first batch at the new site, and we observed a completely different performance. And what was striking, the biggest difference was the quality profile. We spent months troubleshooting. We tested everything. We looked at all the details. Is it the media? Is it the same lot numbers? Is it the C train? Is it the cell banks? Is it the bioreactor control? Even though they're different models, they might have different softwares. Same set points. Are there any differences in the environmental model? Everything was in spec. Finally, after exhausting every rational explanation, someone noticed the not so obvious. We had different light exposure in the two labs. So we ran a controlled study. The light exposure was indeed affecting the culture. Not enough to trigger an out of spec, but enough to change the cell behavior and, more importantly, the product quality at the end of the day. So what is the lesson for us? List all differences up front, and even then expect surprises. And in addition to all the process and analytical parameters, environmental factors matter. Light, temperature fluctuations in the room, perhaps it's even different raw material lots, vibration from nearby equipment. Document everything, even the obvious stuff. Now, let's look at your mass transfer checklist. Calculate scale-down ratios for key parameters. The mixing time increases with scale. You need to plan for that. Power per volume is typically kept constant. The KLA, or in other words, the oxygen transfer, here expect a decrease as you're scaling up. Impeller tip speed is a major consideration for shear sensitive products. Run scale-down models at the receiving site before you commit to full scale. This isn't optional. You need to prove the physics work in their equipment with their utilities, especially if they have different bioreactor models. And document everything, not just the process parameters, the room temperature range, the water quality specs, the equipment maintenance history, the things that live in people's heads and never make it into the batch records. Let's look at the build versus outsource dilemma. And I've heard this question multiple times when I was consulting people, especially smaller companies. When should you build internal? When should you outsource? Let me walk you through. In a nutshell, it depends on your longer term strategy. For instance, if you have a platform technology you will use repeatedly, and if you're a monoclonal antibody company planning 5 programs over 3 years, in-house manufacturing may start to make sense because you will have a core competitive advantage in manufacturing. And if your IP is in the process, not just the molecule, you might need to keep it in-house. But this very much depends on your company. And I also want to say there's no strategy fits all for everyone. It will also depend on the commercial volumes, if they justify your capital investment, and consider also when you're building that you have quite a long lead time and you need to have the expertise to train the manufacturing staff. But I would say for most companies, especially in early stages, it makes a lot of sense to outsource. If you have a novel modality, even if you have a mAb with uncertain commercial potential, if you need speed to clinic, CDMOs have the infrastructure ready today. Also, if you have limited capital for facility investment, outsourcing makes a lot of sense. Or just if you want a strategic redundancy, for instance, you want to test multiple CDMOs so you're not dependent on a single partner. Building internally may make sense if you have a unique cell line, a very unique system where you just don't find the CDMO that has the expertise. Matthias Müllner gave a good insight on one of the podcasts. He said, external CMC expertise bridges gaps early, build internal capability gradually. You don't need to decide everything on day one. Start with outsourced and then learn what you actually need, and then start to internalize what is critical and where you want to build the knowledge in order to create a competitive advantage. All right, let's look at your 12-week tech transfer preparation protocol. We're going to make it very practical. Weeks 1 through 4 will be your foundation. That's when you define your quality target product profile and critical quality attributes with the receiving site. Not at them, with them. That's very important. You start already building the team with them, start to get to know the people. You map the stakeholder landscape using the protocol we just covered. Create a detailed tech transfer plan document. This becomes your contract with yourself. And identify which parameters are scale dependent versus scale independent. Then in weeks 5 through 8, you focus on the risk mitigation. Conduct a gap assessment. Look at the equipment, the methods, the training needs. Qualify raw materials at the receiving site. Source vendor approval, the comparability, because you cannot assume your supplier has the same qualification status at their site. Run scaled down models. Develop a comprehensive sampling plan with statistical power. Train receiving site personnel, and I don't mean train them on SOPs, train them on the why, and if you can, go there, send some staff over to the receiving site and train together. Because this step matters a lot. Because what happens if it goes wrong? What are the early warning signs? You need to prepare for that. And here is the critical one. Document tribal knowledge. Capture the undocumented tricks, the environmental nuances, the operator expertise that lives only in people's heads. The way your senior operator knows the culture is stressed before the dissolved oxygen trace shows it. Write that down. In weeks 9 through 12, you want to focus on the execution preparation. Co-author batch records with the receiving site. Not you write, they review. Co-author. Establish communication protocols. For instance, who calls whom when something goes sideways. Define success criteria and go and no-go decision points. And plan for contingencies. If the scale-down model fails, what's plan B? Let me give you some no-go decision points. You don't proceed to full scale unless the scale-down model achieves comparable results. All CQAs have validated analytical methods at the receiving site. Key personnel are trained and can demonstrate understanding. Raw materials are qualified and available. If any critical gap remains, you stop. You do not proceed to full scale hoping it will work out. Hope is not a strategy. Early in my career, I thought I needed to control everything. Write all the protocols, review all the data, draft all the reports. What was the result? Well, I became the bottleneck. I burned out and paradoxically, progress slowed. The turning point came when I got an assistant for admin and protocol and report writing. Suddenly, I had time for the things only I could do. The strategic decisions, the stakeholder relationships, and the critical technical judgment calls. And this is the insight that changed everything. As a leader now, especially also after my entrepreneurial season, I focus on the 20% that only I can do. And I want you to encourage, start focusing on the 20% that only you can do. You have limited hours and limited attention span. If you spend them on the 80% that someone else could do, you're stealing from the 20% that creates actual value. Here is an action item for you this week. Identify your 80% activities, then plan to delegate them within 90 days. Not someday, within 90 days. You're staring now at your tech transfer timeline. Leadership wants aggressive dates. The CDMO is confident but vague, and you are carrying the weight of knowing that if this slips, then the entire program slips. Do you feel that pressure? Yes, it's real. The complexity also real. But here is also what's real. Tech transfer isn't magic. It's not an art. It's building on 6 essential pillars with discipline and foresight. You've got the framework now. Technical process understanding, in other words, know what's scale dependent. Analytical comparability, design this upfront. Number 3, quality by design. Define your CQAs or your critical quality attributes before the crisis. Number 4 is stakeholder management, where you need to understand what people need. Number 5, your partner selection. Choose the CDMO that challenges you. And 6, strategic build versus outsource. Focus on competitive advantage and speed to clinics. You've got the 12-week protocol, foundation, risk mitigation, and execution preparation. And you've got the mindset shift. Master the human element, understand the physics, plan systematically, delegate the 80% that doesn't require your unique expertise. Your piece of cake tech transfer, it actually can be cake. Not because the tech transfer got easier, but because you got better at orchestrating complexity. The timeline is not going to get less aggressive. The stakeholders aren't going to get less demanding, but you now have a system that works regardless. Go make it happen. If you found this conversation on scale up and technology transfer valuable, I'd appreciate you sharing it with one colleague who is facing their own tech transfer challenge. Sometimes the most valuable thing we can do for each other is share the framework that prevents the million-dollar mistakes. Thank you so much for tuning in today. Thank you for listening. Thank you for leaving a review on your favorite podcast platform, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.